Good day, class. Are you all good? I hope you're all doing good. So, before I start my lesson, let me introduce myself. I am Lena Rose Daisy A. Gabuya, your science teacher. So, let me ask you to bow your head to start our prayer.
Good morning, Lord. Today's a new day. A chance for a new start. Yesterday is gone. And with it, any regrets, mistakes, or failures I may have experienced. It's a good day to be glad and give thanks. And I do, Lord. Thank you for today. A new opportunity to love, give, and be all that you want me to be. Today I want to start the day with you on my mind and in my heart. As I dress, let me wear the armor you've provided daily, the helmet of salvation, the breastplate of righteousness, the shield of faith, the belt of truth, the shoes of peace, and the sword of the Spirit, with prayer on my tongue, praise for you, and petitions for those around me and those I meet. Feed me today with your daily bread, as the bread of life. Your food, like manna, will sustain me throughout any trials and hungers. Help me to set my thoughts on things above, and to speak only what will help and encourage others. Keep me from putting my foot in my mouth, and help me guard the affections of my heart today, Lord. Make whatever work I do be marked with excellence rather than perfectionism. As I seek not to make a name, but to make a difference. Help me to treat each person I encounter as you would, with respect and love, forgiving others and asking for forgiveness myself when needed. As I start this day, help me remember that I belong to you, and my desire is to act accordingly. Keep my feet from stumbling, and my mind from wandering into distractions that could steal precious time and energy from the most important things you have designed for me. I'm proud to be your child, Lord. And I'm so grateful that you died for me, rising again so that every day could be filled with the wonder of your love, the freedom of your spirit, and the joy of knowing you. I know earthly life is short and fleeting, Lord. But I want to live today as if it were the first or the last day of my life, giving thanks for every good and perfect gift you choose to give today and every day. I want to live my life for you, Jesus. In your precious name, I pray. Amen.
So now, I prepared a riddle for you, class. Who do you think gonna get the first question? And who do you think gonna get the perfect score? Let's see. Okay, congrats to those students who get the perfect score. And now, let's talk about our topic or the discussion for the day, which is about the first topic is about genetic engineering. Genetic engineering. So, what is genetic engineering? So, the genetic engineering is artificial manipulation or alteration of genes. Genetic engineering involves of removing a gene or the target gene from one organism and inserting target gene into DNA of another organism, cut and paste process. Genetic engineering is a process by which pieces of DNA are transferred from one organism to another, like human to bacterium, bacterium to plant, human, sheep, and fish to plant. And here's the example. The tobacco plant glows in the dark in 1986 because the gene taken from firefly are injected into the tobacco plant. That is why it glows in the dark. So GMO or the genetically modified organism. Some important terms that we need to know like recombinant DNA is the altered DNA. Genetically modified organism is GMO is an organism with an altered DNA. So the genetic engineering breaks the species barrier. Genetic engineering allows DNA from different species to be joined together. This often results in a combination of DNA that will never be possible in nature. For this reason, genetic engineering is not a natural process. If DNA is transferred from one species to another, the organism that receives the DNA is said to be a transgenic. Example of cross-species transfer of genes. A human gene inserted into a bacterium, a human gene inserted into another animal, and bacterial gene placed in a plant. So the tools used in genetic engineering first is the restriction enzymes. Are special enzymes used to cut DNA at specific places. Different enzymes cut DNA at specific base sequences known as recognition site. Like for example, one restriction enzyme will always cut DNA at the base sequence like GAATTC, and another restriction enzyme only cuts at the sequence of GATC. If DNA from two different organisms is cut with the same restriction enzyme, the cut ends from both sources will be complementary and can easily stick together. So here the restriction enzymes. And the second one is the ligase or the DNA ligase. Enzyme which act like a glue sticking foreign DNA to DNA of a cloning vector will only work if DNA from the two DNA sources has been cut with the same restriction enzyme. That is, sticky ends of cut DNA will be complementary to each other. So here's the 5 steps involved in this process, like the isolation, cutting, insertion, transformation, and expression. Note that the following example will explain how a human gene is inserted into bacterium so that the bacterium can produce a human insulin. So you must know 3 applications, one involving a plant, one animal, and one for a microorganism. So in the plants, weed killer resistant crops. Many types of crops plant have bacterial genes added to them. These genes make the plants resistant to certain weed killer herbicides. This means that the weed killers kill the weeds but do not affect the transgenic plants. In animals, there is a growing trend to experiment with inserting human genes into the DNA of other mammals. The transgenic animals formed in this way will then produce a human protein and secrete it into their milk or even into their eggs. In animals, the sheep produce human clothing factor. A human gene has been inserted into DNA of a sheep. This allows the adult sheep to produce a clotting chemical needed by hemophiliacs to clot their blood and produced in the milk of the sheep. Farming is the production of pharmaceuticals by genetically modified animals, that is sheep, cows, goats, and etc. So in the microorganisms, the bacteria make insulin. The human insulin gene has been inserted into a bacterium or the E. coli. This allows the bacterium to produce insulin for use by diabetics. In ethical issues in genetic engineering, the release of GMOs into the environment, danger of possibility of GMOs being released into the environment. GMOs grown in bioreactors do not provoke as much fear. GMOs grown outdoors alters fears of foreign genes they contain being spread to other plants. And GMOs as a food source. Outlined below are some fears associated with the use of GMOs as a food source, like cannibalism. Eating an animal containing a human gene is a form of cannibalism. Feeding GMOs containing human genes to animals that will later be eaten by humans. So religious reasons. Eating cow genes that are inserted into sheep will be offensive to Hindus. Offensive to vegetarians or vegans. Eating an animal genes contained in food plants cause concern. In animal welfare, there is a serious concern that animals will suffer as a result of being genetically modified. Use of growth hormone may cause limb deformation and arthritis as animals grow. So, uses of genetic engineering is to make growth hormone to treat dwarfs, to prepare vaccines, and to make plants resistant to disease, and to make pigs, cows, or fish grow faster.
Many years ago, diabetics who needed insulin to survive had to inject cow or pig insulin into their bodies. This was not ideal for many reasons. Today, the insulin that diabetics inject is human insulin, and it is produced by microorganisms such as the E. coli bacterium and certain strains of yeast. But how do these microorganisms produce human insulin? Genetic engineering. Scientists are able to turn certain microorganisms into mini factories that make useful substances that improve our health, the environment, and the economy. But what is genetic engineering? An organism's DNA makes up its genes, which code for all the proteins the organism needs to survive. Each gene codes for a different protein, or part of a protein. Genetic engineering is the manipulation or changing of the DNA of an organism. Genetic engineering involves removing a gene from one organism, which is called the donor, and transferring it to another organism, which is referred to as the recipient. The recipient is called a transgenic organism or a genetically modified organism. There are two basic purposes of genetic engineering. Sometimes we require large volumes of a particular protein to be made. Scientists use transgenic microorganisms to produce large volumes of this protein. For example, insulin, growth hormone, or vaccines. An organism, B, can have a gene from a different organism, A, introduced to give it, B, some advantage that A has naturally. Organisms like B are called genetically modified or transgenic organisms. For example, a gene that manufactures a toxic chemical in a bacterium can be introduced into a maize plant to make the plant toxic to certain insects that would eat it, such as caterpillars. Let's now look at the process of genetic engineering. How do you transfer a genetic characteristic from one organism to another? Although it's a very complex process, it can be summarized rather simply. we'll use the example of how the insulin gene is inserted into a bacterium in order to manufacture large amounts of human insulin. The gene that codes for human insulin is found on chromosome 11 at position 15.5. The insulin gene is cut from chromosome 11 using special enzymes called restriction enzymes. Bacteria possesses small circular strands of DNA floating in their cytoplasm. These strands of DNA are called plasmids. A plasmid is extracted from the bacterium cell. The plasmid is cut open using the same restriction enzymes that were used to cut the insulin gene from the human chromosome. The restriction enzymes leave sticky ends, where one of the two DNA strands is slightly longer than the other. Because the same restriction enzyme was used to cut both the human DNA and the bacterial plasmid, the sticky ends are complementary and will allow for joining using complementary base pairing. The insulin gene is joined to the plasmid by the sticky ends. An enzyme called ligase is used to complete the joining of the two pieces of DNA. The modified plasmid is then reinserted into the bacterial cell. The modified bacterium is a genetically modified or transgenic organism because it contains some human DNA along with its own bacterial DNA. Its bacterial DNA is called recombinant DNA, bacterial DNA recombined with human DNA. The bacterial cell is placed in a fermenter, which allows for rapid asexual reproduction in ideal conditions with the optimal temperature, pH, and lots of food. Because the bacteria reproduce asexually, the offspring are all clones of the original transgenic bacterium. They all have identical recombinant DNA. The bacteria express their own genes in order to survive. In other words, they make their normal bacterial proteins. But they also express the human insulin gene and make human insulin. The fermenter has become a culture of mini factories, all producing human insulin. The insulin is then extracted, purified, and packaged. Humans can now inject themselves with human insulin, and that is how genetic engineering works. What is DNA fingerprinting or DNA profiling? Leicester University geneticist Alec Jeffreys developed a technique called DNA fingerprinting in 1985. It allows DNA samples from different people to be compared to look for similarities and differences. It's used for solving crimes and can also confirm if people are related to each other, like in paternity testing. Any two people in the world have 99.9% of their DNA the same, so this process analyzes the differences in the remaining 0.1%. This modern technology is called DNA profiling. It's a very sensitive technique which only needs a few skin cells, a hair root, or a tiny amount of blood or saliva. There are sections, or loci, of chromosomes, where instead of a gene consisting of a long sequence of bases, there are much shorter sequences of 3, 4, or 5 bases that are repeated many times. For example, these repeated sequences are called short tandem repeats, or STR. At these places on the chromosomes where we find these STRs, there are areas that vary in number of repeats. DNA profiling only looks at these STRs. A cell sample is collected. This could be from some blood at a crime scene, or a swab from the inside of someone's cheek, for example. The DNA is then extracted from the sample. Many copies of this DNA may be made using the polymerase chain reaction, or PCR. Special enzymes called restriction endonucleases are used to cut the DNA up into different size pieces. The DNA samples are then put into wells in a special gel called agarose for the process of gel electrophoresis, which separates the DNA fragments by size. The pattern is then transferred to a nylon sheet in southern blotting. And finally, the lines produced by the DNA samples from different people are compared. So let's have a go at using DNA profiles to solve some mysteries. An item was stolen in a burglary. a drop of blood was left behind by the thief. Samples of DNA were taken from 4 suspects and compared to the sample left at the crime scene. Which suspect is guilty? Here's a clue. Look for the one that is most similar to the one from the crime scene. Pause the video while you decide. Suspect 3 is guilty. Can you see how the pattern of bands matches in Suspect 3 and the crime scene? Their DNA is the same. Let's try another. A soldier has been killed in an explosion and has lost his dog tags that identify him. 3 soldiers are missing from their unit, so the army asks the 3 sets of parents for a DNA sample so that they might compare it to the soldier's DNA and therefore make an identification. Remember, the soldier will only share half of his DNA with each parent. So which set of parents is the soldier the son of? Pause the video while you decide. Parents C and D. As you can see, he inherited the first band from parent D and the second, third and fourth from parent C, and so on. So now you understand how DNA profiles or fingerprints are made and seen some examples of where they can be used. You can even interpret a DNA profile.
So let's say that you want to create a custom-made genetically engineered designer baby from scratch. You'd want your baby to be as smart as Albert Einstein, as attractive as George Clooney, and as strong and muscular as The Rock. But you would also want your baby to resist diseases and live for as long as possible. Intelligence, beauty, strength, and life expectancy are all controlled by our genes, which means you could add all these characteristics into one individual before birth. The first thing you need is a skin cell, which can be converted into a human fetus. There are around 35 billion skin cells in your body. It is astonishing to know that each individual cell has the potential to become a fully grown human being. First, you take skin cells. They could be from a male or a female. One person is enough. Then you convert them into embryonic stem cells. These embryonic stem cells have the capacity to become any other type of cell in the body. Heart cell, lung cell, liver cell, neurons, and the list goes on. And since we're interested in creating a human being, we would convert these stem cells into sperm and eggs. This can be done through a process called cellular reprogramming, and it works by feeding stem cells the right ingredients of hormones and growth factors, and sometimes a bit of genetic modification. Depending on the combination of hormones and growth factors that you choose, you can force stem cells to become any type of cell you want. Now that we have converted skin cells indirectly into sperm and eggs, we can fertilize the egg using the sperm through in vitro fertilization, a standard procedure that is currently used by millions of infertile couples around the world, and it has existed since the 1970s. In vitro fertilization allows us to create embryos from sperm and eggs under lab conditions. Unfortunately, these embryos are susceptible to genetic diseases. They could mutate and eventually experience serious health problems after birth. They may also give rise to ordinary human beings. To solve this problem, we will have to genetically engineer these embryos using CRISPR-Cas9, a gene editing tool that is fast, reliable, and precise at editing the genes of almost any cell. There is a big difference between engineering an embryo and engineering an adult human being. You can't become a Spider-Man simply because a spider injected you with its genes. Genetic engineering works best on embryos because the number of cells is very low and you can edit all of them with minimal efforts. There are simply 4 to 8 cells. However, adult humans already have trillions of cells, 37.2 trillion cells to be precise. Editing all of them at once with our current technologies is near impossible, and it probably won't even be achievable for many decades to come. The other advantage of editing human embryos is that it's a more efficient process. That's because genetic information can pass through generations. If you genetically engineer an embryo to become someone intelligent, the intelligence will pass from one generation to the next. Assuming you did this to an adult, which is impossible for now, the intelligence would remain with that particular individual and will die as soon as he or she dies. That's why we should work with embryos, because any genetic traits introduced here will affect the entire species for a very long term. When it comes to genetic engineering of human beings, people are mainly interested in treating genetic diseases. There are over 6,000 genetic diseases. Our genetics contribute to a number of diseases such as Alzheimer's, arthritis, diabetes, cancer, obesity, and heart disease. The more we learn about the genes that cause them, the greater the chances of fixing them through genetic engineering. As of now, scientists have managed to use CRISPR gene editing to correct genes linked to blood disorders, HIV, muscular dystrophy, and a heart condition called hypertrophic cardiomyopathy. Apart from fixing genetic diseases, you could also engineer the phenotype, which is the external appearance of the person: hair color, eye color, skin tone, and body strength. All of these traits are fully customizable because they are controlled by our genes. And since different people have different standards of beauty and color choices, you could eventually engineer your baby to be the absolute perfection that you're looking for. You could even extend the lifespan of your baby and even enhance human intelligence. We already know that there are over 1,000 genes that are linked to human intelligence. We have pinpointed them, and controlling them will allow us to enhance human intelligence on every born designer baby. In 2018, China became the birthplace of the world's first genetically engineered humans. A Chinese scientist announced that he had created the world's first genetically engineered babies by modifying human embryos using CRISPR gene editing tool. With the help of CRISPR, he introduced HIV resistance to human embryos and then implanted these embryos into a mother who gave birth to 2 genetically modified humans. This triggered a lot of discussion about the ethical issues of such technology. This kind of technology raises ethical questions about who will have access to it and whether it's a form of playing with nature that would backfire against us later on. One thing for sure, this is going to be expensive. In 1996, Dolly, the first cloned animal, was born. Even though cloning is basically copying the genome without any genetic modifications, it was still hard. Out of 277 clones, only 1 was successful. Now think about genetic modification of several genes. You would need tons of embryos to be engineered, and then screen them to find the perfect one, which tells you how costly that could be. Just like with every other technology that existed or will ever exist, there are upsides and downsides for designer babies, some of which can be very hard to predict, especially when talking about our genome, which is constantly changing. Our path to a society where designer babies exist is progressing faster than you think. And with these rapid changes, someday you will wake up and find yourself in a completely different world.
With all the work to do, people to see, traffic jams to get stuck in, we all sometimes wish we could be in 2 places at once. We've already cloned sheep, cows, even monkeys. What if we cloned ourselves? Would a clone have feelings and be self-conscious? Could you genetically modify them to become better than yourself? This is What If, and here's what would happen if you cloned your body. Cloning is reproducing a genetically identical copy of an organism. Scientists are doing that all the time. But they aren't cloning humans. They're mostly making copies of cells or DNA fragments for research purposes. Occasionally, they'll clone an animal too. In the case of Dolly the sheep, scientists took a somatic cell from an adult sheep and inserted it into an egg that had all of its DNA information removed. Since then, we've cloned dogs, cats, horses, and just recently, primates. It looks like researchers have figured this cloning thing out. What if one day they decided to start cloning humans? It turns out some organisms are harder to genetically duplicate than others. Dogs are harder to clone than cats, and rats are even harder to clone than dogs. Humans are also among the toughest. Tough, but possible. What if you were the first one ever to make a clone of yourself? Would it be an exact copy of you? I hate to break it to you, but not only would your clone have a different personality, but they'd also look different. Think about it this way, just how identical are identical twins? You may not notice at first glance, but when you look closer, one of them has a birthmark on their cheek, and the other one is a little taller. While you won't notice much of a difference in a sheep clone, once you looked at your fresh from the lab you'd see that it's, well, not you. You're more than just a product of your genetic code. Your character, behavior, even your appearance depend on the environment in which you were raised. You can make a copy of your genes, but copying your memories and experiences is impossible. Oh, and you realize that your clone wouldn't be your grown-up self, right? You'd have to raise that cloned baby before they got to your age, if they ever do, because clones are typically very unhealthy. Most likely, they'd have a defective heart, liver, and brain, as well as a very weak immune system. Many cloned animals had their cells age much faster than normal. Your clone's body would probably get old and deteriorate much sooner than you. Unfortunately, your clone would be very sick and die early, and they may not even look like you, not to mention a totally different personality. Human cloning is illegal almost everywhere on Earth. Now and then, a group of researchers will claim to have cloned a human successfully, but with no scientific information to prove their claims, human cloning remains in the realm of science fiction. So, if you're ever on a trip abroad and meet someone who looks like you, don't panic. The chances that you were cloned are close to zero. Have you seen your look-alike? What if everyone in the world had a double? Well, that's a story for another What If.
And that's the end of our discussion for today. Class, please study all things that we have discussed today because I'll give you an exam tomorrow. It's just a multiple choice, so don't be worried, okay? Goodbye.